Go to Sherwood. So we're talking about the ganglia, and the individual ganglia as shown here from the anterior view. We've got celiac ganglion and a celiac plexus associated with that. You may have heard the term solar plexus. It's kind of a vernacular term. Boxing, they use that. Oh, he got hit in the solar plexus, or the wind got knocked out of him. That is a series of nerves that also help to, they go through the diaphragm and then go back to the diaphragm. So if you hit this plexus, kind of knocks out the diaphragm from being able to help you with the breathing, the elongation of your thoracic cavity. So it's hard to catch your breath. So I just thought I'd throw that in. Next one will be the superior mesenteric, and then the inferior mesenteric ganglia. We don't really have a named plexus between the superior and inferior. We have an inferior mesenteric plexus, but nothing in between there, but there is a plexus there. So it's just, I guess, a combination of the celiac, the superior, and then down to the inferior plexus. They're all connected together, and the nerves are intertwining together. Below the inferior mesenteric plexus, we have a hypogastric plexus. So it's probably named for the region. If you remember the anatomical regions that we covered in the first chapter last semester, you have epigastric, umbilical, and then hypogastric regions. And so this plexus is in the hypogastric region, so that's probably why that is named hypogastric. You might try and translate that and think, well, it says under the stomach, and that's pelvic cavity, but this is the hypogastric region. So from the side, we can see the celiac ganglion, the superior mesenteric ganglion, and then the inferior mesenteric ganglion, and all the nerves intertwining and then going down to hypogastric. Other plexuses that are not associated with collateral ganglia, or the collateral ganglia are all below the diaphragm. We have up here, we've got a cardiac plexus, and that will have the vagus running through it and also sympathetic nerves running through it. That will go to excite the heart. The vagus will be your parasympathetic input to the heart. Vagus will also go through the cardiac plexus and then go into the pulmonary plexus and then down into the esophageal plexus. So it's helping us with our airways. It will constrict airways when we're not needing to bring a lot of oxygen into our lungs for exercise purposes. So when you're resting, then your airways narrow. They don't narrow to inhibit air, but they narrow because you're not requiring a whole lot of air to come in for oxygenation. It also helps with coordinating swallowing, and then it will also be involved in the GI, the rest of the GI system as well. So visceral reflexes. These we don't see, but they occur in control of the autonomic nervous system, so under our subconscious control. They will modify, and they will be modified by higher centers. All the reflexes are polysynaptic. We had talked about monosynaptic, which aren't truly monosynaptic in relationship to the knee-jerk reflex, because that does involve interneurons, so it's really not monosynaptic, but textbooks describe it as monosynaptic. Here you can just consider everything polysynaptic, meaning there's going to be interneurons and probably multiple neurons involved in the reflex. They're divided really into two groups. Does the central nervous system get involved in the reflex? If it does, then it's a long reflex. If the central nervous system does not get involved, and only the ganglia are involved in the reflex loop, then that's a short reflex. So long reflexes, the afferent will go through the dorsal root ganglia, will go into the spinal cord, and then it's going to, if it's a cranial reflex like tearing or blinking when something gets into your eye, then it's going to be efferent with cranial nerves. If it's down below, then it's going to be efferent with the autonomic nerves, the motor nerves of the autonomic nervous system. So it's either going to be integrated in the brain stem for your cranial reflexes, or it's going to be integrated in the spinal cord for your visceral reflexes. When you have a long reflex, then you have a greater chance for divergence. Short reflexes, you're not going to have as much divergence because you're limited with the number of cells that you can innervate with what is available in the ganglion itself. So they're actually processed in the autonomic ganglia, and then they're going to have postsynaptic neurons that will be the motor neurons that will command the effector organs. These short reflexes that are integrated in the ganglia, they're going to go back to very discrete parts of an organ. The long reflexes, when they recruit many more postganglionic neurons, they're recruiting first the preganglionic neurons, and then they're recruiting the postganglionic neurons. You get a much more widespread effect of the reflex. So short reflexes, this is one part of one organ that's going to be responding. So here's the figure that they've drawn. If it's going to be a long reflex, then the cell body of that afferent neuron will be located in the dorsal root ganglion. It will go in through the dorsal horn and then into synapse with interneurons. These interneurons may recruit multiple preganglionic neurons, and then they in turn recruit multiple postganglionic neurons. If you are bypassing the central nervous system, then the cell body of that neuron is going to be located outside again, but not in a specific area, not in a ganglion. So no collection of those cell bodies. They're just located close probably to their organ, and then a longer axon going to the ganglion and then interacting with interneurons and recruiting postganglionic fibers. These fibers then go back to a very discrete area, probably that same area where the sensory neurons originated, to have their effect. Hypothalamus, posterior and lateral hypothalamus is going to control sympathetic input to your autonomic nervous system. So although most of the chapter is focusing on your pre- and postganglionic neurons, you have to remember that the hypothalamus is an integration center, and it's not acting alone. It is influenced by higher centers. It is influenced by your limbic system. So there's many inputs to the hypothalamus, and then the hypothalamus is going to communicate with the medulla, and then the medulla is going to communicate with your preganglionic neuron. So there's a lot of things going on above the level of your preganglionic neurons in your spinal cord. The part of the hypothalamus that is collection of sympathetic cell bodies or sympathetic nuclei will be most in the posterior part of the hypothalamus and posterior lateral, so sides. And then parasympathetic is on the opposite side. It's anterior and medial hypothalamus. And it too will influence the brainstem, and then the brainstem will, for the cranial nerves, that is where the cranial nerves exit. 3, 7, 9, and 10 exit from the brainstem. But there will also be long axons that will go all the way on down to the sacral cord and then influence the sacral nerves of the parasympathetic nervous system. A lot of Chapter 7 is repetitious. You can use it for your own review. Chapter 7 actually has some good study tools in it for you, even for the anatomy part of the lectures. There are a few corrections too that I'll be making today in lecture. I will not post them, so if you know somebody who's not in lecture, you can share the corrections with them. So review here, we've got preganglionic located in the central nervous system. Could be brainstem for craniosacral parasympathetic, or in spinal cord for the thoracolumbar and the sacral part of the parasympathetic. Preganglionic axon is going to go to a collection of cell bodies located somewhere. It could be in a very specific autonomic ganglion that sits on top of a blood vessel, and so you can identify it. Or it may be in the head and it's close to the target organs, the salivary glands, the eyes, lacrimal gland, etc. Or it could be in the wall of the target organ, which is the majority of your parasympathetic postganglionic cell bodies. So preganglionic neurons will release acetylcholine. Every single preganglionic neuron, both sympathetic and parasympathetic, release acetylcholine. So we have a cholinergic receptor that will then stimulate this postganglionic cell. Action potentials travel down. The difference between a postganglionic neuron and a somatic motor neuron is both where your cell body is located. In the somatic nervous system, it's back here, and you've got one axon going out to your target organ. Here we've got two steps. We've got pre and then postganglionic. The postganglionic terminal also is different. Instead of having little branches with individual boutons and then individual neuroeffector junctions, we've got this wide branching series of branches that each branch has many, many little swollen areas called verrucosities that contain the neurotransmitter. So they can release neurotransmitter over a very widespread surface area. So they can hit multiple synapses, not on just one particular cell, or motor unit, but over many, many cells. The neurotransmitter has to be either reuptaken by those verrucosities to be destroyed, Otherwise, if it's not, it diffuses out and can get into the blood and then will not be destroyed till it gets to the liver or the kidneys. So widespread effects in that way as well. This figure here is not so good, especially down here at the bottom. The cranial and sacral division, they have it color-coded as parasympathetic. Parasympathetic terminal ganglia are all in the head. There are no terminal ganglia in the sacral part of the parasympathetic nervous system. These are intramural ganglia. So you need to make that change. And this doesn't really exist far away. It's actually within the target organ itself. So this, you got to erase that ganglia and actually put it into the target organ. The rest of the upper part is okay. You got cranial, you got cranial nerves. They go to terminal ganglia. Postganglionic cell goes to the target organ. So you'll have your lacrimal glands, some of your salivary glands, and your pupil and lens constriction, taste, and blood pressure regulation, all via cranial nerves. The sympathetic division, you have short preganglionics that synapse directly in the paravertebral chain of ganglia, and then a long unmyelinated neuron that will release norepinephrine. You've got a pretty long myelinated neuron that will go directly to the adrenal medulla and then release the norepinephrine, or about 15% of that, and then the rest will be the 85% epinephrine that will go into the blood. The epinephrine and norepinephrine can bind to that same receptor that the nerve would have released norepinephrine into that area. So from the bloodstream, you can stimulate that receptor. You can also stimulate receptors that are specific or have a higher affinity for epinephrine, and you have some that really only bind epinephrine. These are your beta-2 receptors. Now, beta-2 receptors, they stimulate adipose. That's another problem I have with this figure. This implies with this direction and the adipose tissue being right lateral to that postganglionic neuron of the parasympathetic, that it's recruiting adipose. It's causing lipolysis. Parasympathetic does not do that. The sympathetic nervous system via the beta-2 receptors will cause lipolysis. So actually parasympathetic doesn't do anything to adipose. So a better figure is coming up, 7-3. I would just kind of use this central part for review. Not so bad up here either, just missing the pictures of the target organs. But down here, definitely some problems that the authors need to work out. This is a good review for the test. What is acetylcholine? What is its function as a neurotransmitter? Well, all preganglionic, whether it's parasympathetic or sympathetic, is going to release acetylcholine. So all the postganglionic cell bodies will have cholinergic receptors. All parasympathetic postganglionic effector receptors or terminal receptors are going to bind acetylcholine. So these postganglionic cells release acetylcholine. The parasympathetic nervous system is easy. It's ACH, pre and postganglionic. The sympathetic postganglionic receptors terminate at sweat glands. Now there's some debate, I'm going to talk a little bit about it towards the end of this chapter. Is the sympathetic nervous system the only thing that is in control of all our sweat glands? For sure, when we get into a hot environment, that cooling type of sweat, that's sympathetic involvement. Sweat due to exercise, sympathetic involvement. But what about nervous and anxious sweat? We'll talk about that later. But for the sweat glands that are under sympathetic control, the major ones that do the major sweating for cooling, that is, those sweat glands will be stimulated by acetylcholine released from sympathetic neurons. The skeletal muscle, we learned last semester, under the control of acetylcholine from the alpha motor neurons. And then certain areas in our central nervous system also under the control of acetylcholine. So our brain also uses acetylcholine to communicate in certain areas. The norepinephrine is primarily coming out of the postganglionic neurons of the sympathetic side of the ANS. So most, that's your qualifying term, most sympathetic postganglionic terminals release norepinephrine. Adrenal medulla is going to release primarily epinephrine, and that's because it has, those cells have all three enzymes to take tyrosine to dopamine, take dopamine to norepinephrine, and then norepinephrine to epinephrine. So they can go all the way down to epinephrine. They do release some norepinephrine because not all of the norepinephrine gets converted to epinephrine before the adrenal gland is stimulated to release. That's fine because norepinephrine binds to all its receptors. It has affinity for most of the other receptors that epinephrine will bind to. And then norepinephrine is also used in our central nervous system, so our brain and spinal cord will use that for communication as well. So this figure is actually better, but the only drawback is some of the terminals are kind of small, hard to see. So you got to look closely. When you see all these little tiny little synapse with a dot, that dot is the cell body of the postganglionic neuron, and that little fork there is the nerve terminal of the preganglionic neuron. When it's a little dot right by a fork, then that is intramural ganglia. So you see that the lower parts of the parasympathetic nervous system is all intramural ganglia. And then here with the sympathetic, they've sort of crunched all the ganglia together, and they're all going through that. There's a little circle here. This might be your superior and inferior mesenteric ganglia. I'm not really clear as to what that is supposed to be. I tried to find some reference in the textbook, didn't. But anyways, you've got the collateral ganglia here, you've got your sympathetic chain of ganglia here, got terminal ganglia associated with the parasympathetic in the cranial nerve side, and then you've got your cervical ganglia for the postganglionic cells on the sympathetic side. Up here in the eye, way, way, way back, I don't know how long ago, but back when Raphael and some of those early painters were drawing or painting beautiful women, it was very popular for women to put belladonna in their eye, which you wonder, like, well, why did they do that? Because isn't that Can't that kill you? Yes, it can. But a small amount that they put in their eye caused an inhibition of the constriction due to the parasympathetic nervous system. So they walked around with these dilated pupils, and that was considered very beautiful. And so the plant actually was named for this beautiful woman syndrome. So Bella meaning beautiful, and Donna meaning the woman. So belladonna, one of the active ingredients in belladonna is atropine. So atropine, now they know that's a good parasympathetic nervous system inhibitor. So remember that, because we'll talk about that when we get back to sweaty palms. Okay, it's not going to want to load.
Oh yeah, it's
Their ganglia usually lie near the spinal cord. Parasympathetic nerve.
Terminal ganglia up in the head on the parasympathetic nervous system side. So sympathetic nervous system doesn't use terminal ganglia in the head. And they said all major organ systems are dually innervated. So remember, all major is your qualification, because you'll see that not everything is dually innervated. So exceptions to dual innervation. Arterioles and veins, they're usually under sympathetic control, not influenced by parasympathetic input at all. Sweat glands, the ones that do the cooling for you in a hot environment or due to exercise, are under sympathetic control. But back in the 1930s, I think somewhere around 1937, they started looking at psychiatric patients, people who were had, and then later on post-traumatic stress disorder, although they didn't call it that at the time, but people who had high anxiety due to war experiences. They noticed that they seemed to have more sweaty palms, even when they were relaxed and not saying that they were feeling any type of anxiety. They had sweatier palms. So somebody had a clever idea to get some paper and put a chemical on the paper that would react to some chemicals in your sweat. And then they had the patients touch the paper, and then they had people who worked in the psychiatric hospitals touch the paper, and then they ranked the amount of sweat. And for the people who were the psychiatric patients, they had excessive sweating, meaning up to, out of 5, they were in the 4 and 5 region. Even when they were not reporting that they felt anxious, they still were in the 3 to 4 categories. And then when they looked at the people who worked with them, who, if you're working with psychiatric patients, you get anxious too. They were in the 1 to 0 range, and sometimes up to 3, but never in the 4 and the 5. So I thought, let's go outside the psychiatric hospital and see what we can find out there. And they got people from the general population, asked them if they were anxious or not, then had them touch the paper, and it seemed to correlate that even though the outside population reported that they felt anxious, their palms were only sweating in the 0 to 1 to 2 and not even into the 3 range. So they were much less sweaty than these people in the psychiatric hospital. So then somebody back up in the 50s decided, let's look at whether we can induce anxiety and see if we can elicit the same type of sweating. So they had people, they inflicted pain in the palm of the hand. They had them do mental arithmetic, and for most people who are mathematically challenged, that's stressful. And then they also had them do static exercise. So not running around, but just exercising in place. And then they also induced thermal stresses on the patient. So they induced thermal stresses on them, but then they had their feet in cool water or some other way of controlling the core body temperature so the core body temperature didn't get elevated, and looked at what kind of sweat rate they had in the other areas. And what they could do by also putting atropine into their bloodstream was stop sweating. So even though they were in an environment that should induce sweating, if they artificially cooled the blood, they did not sweat. If they put atropine into their blood and blocked the theoretically parasympathetic nervous system, they did not sweat. They did this with psychiatric patients who had the 4 to 5 category sweating palms. They gave them atropine and they didn't sweat. So does this mean that parasympathetic nervous system is involved in palmar sweating? Or does it mean that atropine is affecting the postganglionic receptors that are binding acetylcholine from sympathetic nerves? So is it a receptor effect, or is it a part of the autonomic nervous system effect? Anybody have an idea? Got one brave person who says it's a receptor effect.
That's just a guess. I think
Well, neither can anybody else.
Oh, okay.
No, there is no right answer. Nobody knows. A lot of people say since it's atropine that is blocking, and atropine blocks parasympathetic nervous system at the eye, then parasympathetic nervous system must be involved in palmar sweating. Is that logical? Not so, because it could be a receptor effect and only a receptor effect. So we got to look at the receptors and see, are there similarities between the receptors up here that are parasympathetic and the receptors here that may be sympathetic but have the same kind of affinity for atropine and then the same response to atropine. So I've looked up as late as 2013. Nobody is saying one way or the other. Is it parasympathetic or is it a receptor effect? In fact, nobody is saying it's a receptor effect. So do the research. Yeah. Get $1 million grant. No, that's what you do. You write the grant and then you get the $1 million for them to do it. Yeah. Okay, so salivary glands, they're duly innervated, but the secretions are not controlled. They're parasympathetic, watery secretions with high enzyme content, the ones that will help with digestion of the food. Sympathetic is the mucus secretion, and that when parasympathetic is high, then you have a lot of salivation, a lot of drooling. When sympathetic is high, you have dry mouth and sticky mucus in your mouth. So they're innervating the glands, but the different cells in the glands will respond to different parts of the autonomic nervous system. So the autonomic nervous system, if they're both innervating an organ, then they're both turned on to some degree at all times. Never one is turned off and then the other is turned on. So nobody is ever turned off, but the ratio of what their input is to the organ will determine what effects are occurring. So they're both partially active at all times. One is dominant over the other, either slightly or in the extreme. So the sympathetic nervous system is going to stimulate the heart rate, and it's also going to stimulate the force of contraction. Parasympathetic nervous system keeps the heart rate down. It has no effect on contractility. So the first thing you do when you start exercising is the central nervous system dials down the parasympathetic nervous system, and that will allow your heart rate to go up. If you're moderately exercising or just doing light exercise, that may be sufficient to increase your cardiac output and maintain that level of activity. If it's not, then the sympathetic nervous system has to crank up, and then the heart rate will go above 100, and the force of contraction will also increase. Yes?
So let's say if you have an elite athlete whose heart rate is low because he's so athletic, parasympathetic is dialed up more than sympathetic?
That's a very good question. What the autonomic nervous system is doing is gauging how much increase in blood pressure is necessary to maintain the delivery of oxygen to the tissues. The heart rate doesn't go up as much because the force of contraction due to the thickness of the wall of the heart has improved the stroke volume on that side of the cardiac output. So cardiac output is stroke volume times heart rate. So if you have a greater stroke volume, you don't have to increase your heart rate as much. So there is a benefit to aerobic training in that the heart will hypertrophy in a good way and become more efficient at pushing blood out per contraction. And then you don't have to elevate the heart rate as much. Now, whether or not the parasympathetic is dialed up or it's sympathetic is dialed down more, so that there's a greater difference between the normal parasympathetic and sympathetic input, I don't know. Another study for you to do. You're going to be busy. Okay, so the sympathetic in the blood vessels will go to alpha-1 receptors and cause constriction. The parasympathetic will dilate vessels that are supplying the penis and the clitoris. So it's a very specific location for parasympathetic input. The rest of the blood vessels are not under parasympathetic control. Just that part of the arousal in the sexual organs in the clitoris and the penis, those are under parasympathetic control. So the alpha-1 receptors, and then there are also alpha-2 receptors that are in the GI tract. When sympathetic nervous system stimulates those, then you get a shift of the blood out from the GI tract into the systemic circulation. The constriction here will be overridden by the local effects of metabolism. So if you increase metabolic rate around blood vessels that have constricted, then increase in CO2, drop in pH, increase in hydrogen ion, ADP concentrations, temperature, all of those override this sympathetic input. So you get an increase in blood flow despite the sympathetic vasoconstriction. At the lungs, they're dually innervated. Your lungs are going to, the airways will dilate, so more air can come in. This is via a beta-2 receptor. The bronchioles will constrict under the influence of the parasympathetic nervous system. The mucus secretion, they're under influence of alpha receptors. Less mucus comes out into the airways when you want to get the most oxygen into the lungs. Makes sense because mucus will plug up the airways. So you want to get less mucus in there. It's not eliminating mucus secretion, but it's dialing it down so that it's not clogging the airways. The parasympathetic increases mucus secretion. Digestive system has input from alpha and beta receptors, alpha 2, beta 2. These will decrease the motility. When you're exercising, your GI system shuts down. So you don't go swimming right after you've eaten a meal because your GI system will just have solid food in there and the blood will be leaving to go up to your systemic muscles, your systemic capillaries, and you probably get abdominal cramps as a result. The sphincters will tighten, so you get a constriction. So what's in your stomach is going to stay in your stomach. What's in your small intestine is going to stay in your small intestine because there's a sphincter there between the small and the beginning of the large intestine. And hopefully your internal anal sphincter also is constricting so you're not pooping in your pants as you exercise. So secretions go down, so digestion goes down. Everything is under sympathetic input is shutting down in the digestive system. The blood has gone out to your systemic circulation. When you're under parasympathetic control, motility increases, so things move from one section to the other for digestion, for absorption and secretion. Sphincters relax, but they're also under hormonal control, which we'll find out actually has higher control than the nervous system. So if there is a reason not to allow something to come into the next section, then the hormones will be released and those will keep the sphincter tight. So you can have a fight between the parasympathetic and the hormones that are released downstream of that sphincter. The hormones will win out. So secretions will increase, so digestive enzymes from the stomach, that's primarily your protein digestion, that's pepsin, and then from the pancreas, you'll have your carboxypeptidases, you'll have your endopeptidases, you'll have your lipases, you'll have your amylases. And then some major input from the liver, you'll have that under hormonal control and parasympathetic control. The bile is necessary for emulsification of the fats. That means you get them into smaller little droplets, so they stay associated with the water, and then the pancreatic enzymes can attack the lipids and break down the the covalent bonds there. Urinary bladder relaxes under beta-2 receptor influenced by sympathetic nervous system. So you retain the urine, the bladder is being stretched, but there's no initiation of a micturition reflex because the smooth muscle is relaxing. So you'll retain the urine with no urge to have to evacuate the bladder. Under parasympathetic input, the bladder, smooth muscle in the wall of the bladder contracts, and smooth muscle in the sphincters relax, and you get voiding. So this is your opposite effect in the pupil. You get dilation so you can see the danger with the sympathetic nervous system. And when you're relaxing, your parasympathetic will cause constriction of the pupil. The eye for far vision, beta-2 receptors relax the muscles that control the shape of the lens. When they relax, they move away from the lens, and that puts tension via ligaments on the lens. The lens flattens, and then it doesn't refract as much light. So you see far objects very easily. Under parasympathetic input, that's for a near response. So the muscles contract. When they contract, they go towards the lens, which slackens the suspensory ligaments and takes tension off the lens thickens, and now it will refract light more strongly. So objects coming close to your face will be in focus on your fovea. Liver, only innervated by sympathetic nervous system. Makes sense. What is the liver doing for you in a sympathetic nervous system response? Number one, release glycogen that's stored. So break it down, get the glucose out into the bloodstream, and then get the building blocks for new glucose, mostly from fat, and convert the fats into new glucose. So parasympathetic nervous system not involved in the liver at all. And then here, another confirmation of the incorrectness of that 7-2 implication. Parasympathetic is not involved in lipolysis at all. It is sympathetic nervous system acting via beta-2 receptors. Exocrine glands. So if you remember, exocrine means you're going to secrete either onto the surface of the body, or you're secreting into a tube that has access to the surface of the body. So sweat glands are exocrine glands because the tube goes up to the surface of your skin and the fluids come out onto the surface of your body. Your salivary glands go into your mouth, but your mouth is access to the external environment. Your lungs secrete into the airways, which then are connected to your pharynx, which is connected to your mouth. So that's the exocrine gland. Your pancreas goes into the duodenum, which then has access via the anus to the environment. So those are exocrine glands. Pancreas has endocrine glands. We studied that last semester. Those go into the blood. So pretty much that's it. So exocrine pancreas, the secretions will be inhibited when you're exercising or highly stressed out. Digestion shuts down under sympathetic control, and that's via alpha-2 receptors. The parasympathetic will increase the pancreatic secretion release. Sweat glands, they're under the influence of the sympathetic, mostly for the cooling type of effect. And then the specialized sweat glands that are in your armpits, genitals, and palms, maybe palms of the hand, those may also be parasympathetic control. Salivary glands, again, depends on what you're secreting. Sympathetic stimulation, mucus comes out. Parasympathetic, the enzymes and the watery secretions come out. So they're going to the same glands. The parasympathetic predominates in the parotid, but 50-50 in the submandibular glands, and sympathetic primarily in the sublingual glands. If you look at the secretion, primarily it's the serous secretion from your parotid, 50-50 from submandibular, and then primarily mucus from sublingual. Endocrine glands, adrenal medulla and endocrine pancreas. Adrenal medulla is totally sympathetic nervous system. The postganglionic neurons are what release the epinephrine and norepinephrine. So they are totally on the sympathetic nervous system side. There is no target organ in the adrenal medulla. It's somewhere else out in the body. So a parasympathetic will have no influence at all on the adrenal medulla. The endocrine pancreas depends on what you're releasing from the endocrine pancreas. The insulin is there to get in your bloodstream to get sugars into the cells, to keep your blood sugar lower after a meal. So it will be released under parasympathetic input. And glucagon is also going to be released under parasympathetic input. Insulin is inhibited by the sympathetic nervous system, and in addition to the slight stimulation of glucagon by parasympathetic, there's a strong influence under the sympathetic nervous system via those alpha-2 receptors. Ejaculation is controlled by the sympathetic nervous system. Erection is controlled by the parasympathetic nervous system. So there are different effects on the same organ. And brain activity, alertness increases with your sympathetic nervous system response. And parasympathetic, at least as far as you know, no real influence on that. Okay, take a break. Muscarinic receptors, we talked about that in Martini's chapter. The nicotinic are responding to nicotine. That means that molecule can bind to the receptor. That's what they're named for. But actually in the body, the primary stimulus is going to be acetylcholine. But interesting, it's because of that nicotine, then when you're a smoker, then you have nicotine binding to some of these receptors as well. And so your autonomic nervous system can also get screwed up by cigarette smoke. Not only lung cancer and stuff like that, but your autonomic nervous system can be increased in its activity in certain areas because of that response. So all autonomic postganglionic fibers will have these nicotinic receptors on them. The preganglionic will release acetylcholine and bind to the cholinergic receptor. And they'll open nonspecific cation channels. So you're opening a cation channel, that means positive charges are coming in or positive charges are going out. Positive charges come in, the cell depolarizes. If positive charges go out, the cell hyperpolarizes. So by opening different channels, you can get two different types of effects. The fact that they're mostly excitatory means that the majority are allowing positive charges to come into the cell, and that's depolarizing the cells. Acetylcholine on the motor neuron, that was last semester. So you can kind of ignore this for this semester. Muscarinic, the acetylcholine will come from the parasympathetic postganglionic fibers. And those will be targeting the receptors in heart muscle, smooth muscle, and then most of the endocrine and exocrine glands in the digestive system, and mostly digestive system, but also respiratory system as well. The mechanism of action for muscarinic, as we said in the Martini chapter, is not ionotropic. It's not opening channels. Ions are not moving. It's stimulating a G protein, and then you're triggering an amplifier enzyme. So the response due to the muscarinic is amplified due to intracellular second messenger system. The amplification due to the nicotinic is due to divergence. So small effect, divergence, but a large excitatory effect intracellularly. The second messenger system can also open ionic channels. So having a metabotropic receptor, you got two bangs for your buck. You got the second messenger system that amplifies the internal response. You can also open channels that can influence the depolarization of the cell membrane as well. You can have two types of effects. Alpha 1, alpha 2, beta 1, beta 2. In general, think of alpha 1s or beta 1s as being excitatory. So that's what the plus sign means. You stimulate something. The alpha 2 or beta 2 usually is going to be inhibitory. So I put these words here because the text does use them and it does qualify. It says that for alpha 1, over here in the table it says it's excitatory, but in the text it says it's usually excitatory. So in other words, it can be inhibitory as well. And down here, beta 2 is generally inhibitory. So in other words, in some areas it can be excitatory. So you should never take anything absolutely positively one way or the other. There's always going to be exceptions. And even if in this area they haven't qualified the inhibitory or excitatory, keep in the back of your mind at some point, maybe Luis will find that it is also, it's just generally or partially. He's going to be doing a lot of research, so he's going to be changing these textbooks for years to come. Okay? And back when I was an undergrad, a lot of things have changed now. What they told you absolutely, they used to teach you dogmatically, absolutely excitatory. And then you get into grad school and you find out it's actually mostly not excitatory at all. And I used to really get my, why do you say it's in one class, same instructor, dogmatically? And then, and then in the graduate school, no, not really. Be honest. Oh, no, we don't want to confuse you. You will when you get to grad school, so might as well confuse us early. Okay, so here, alpha 1s, alpha 2s, beta 1s, beta 2s. What are they responding to? They respond pretty much to both norepinephrine and epinephrine, but the receptors will have a higher affinity for one or the other. And that's what this is about. So alpha 1s are going to have a greater affinity for norepinephrine. You're going to find alpha 1s opposite the nerve terminals. So it makes sense. If the nerve is releasing norepinephrine, you want a receptor that has a high affinity for it. You don't want to have a low affinity for it unless you just don't want to respond to the norepinephrine. So it does have a greater affinity in the alpha 1 receptor for the norepinephrine, and so does alpha 2 have a greater affinity for norepinephrine. Where do you find these? They'll be in most sympathetic target tissues, alpha 1, digestive tract, primarily alpha 2. The beta 1s, they are things like the heart. Sorry about that. The heart, you want it to be able to respond directly to the sympathetic nervous system. So when your brainstem stimulates your cardiac center, your heart increases, force of contraction, and heart rate. But you want to also be able to respond to the circulating hormones that are in the system after a initial sympathetic response. So the nervous system doesn't have to keep on firing because now hormone is arriving. So it may